Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about the anatomy of a dicot stem or internal structure of a dicot stem. Okay, so it is the diagrammatic representation of a typical dicot stem, the transverse section of sunflower stem. Magnified image of transverse section of sunflower or Helianthus anus stem. So, the transverse section of any dicot stem reveals the following layer. The outer layer is the epidermis. It is the outermost layer. It is made up of compactly arranged elongated parenchymatous cells. Which look rectangular barrel shaped in transverse section. It is transparent in nature. By morphology it is transparent. It have no color because it lacks chloroplast. The outer wall are convex, thickened and cutinized. That means it have cuticle layer. On the outer side they possess a layer of cuticle. The internal wall of epidermal cells are thick. So outer walls are thick and inner walls are thin. The radial walls are thick towards the outer side and thin towards the inner side. Gradually become thin towards the inner side. At some places, epidermis contains minute pores called as stomata. Each stomata have guard cells, kidney-shaped guard cells, and those guard cells have chloroplast. And they controls the opening and closing of the stomatal pore. Okay, by their swelling, two guard cells can form a pore in between them, or they can close. So they control the various functions of epidermis. Is they protect the internal tissues, they prevent the entry of harmful organisms, they minimize the surface transpiration because they have cuticle. Because cuticle is oily layer, more or less similar to a oily layer, exchange of gases through stomata. It protects the inner cell from excess heating or sudden change in the temperature with the help of hairs or stomatal hairs, as you can see in this structure, shoot hairs. Next, inner to epidermal layer we have cortex. So it is called as cortex, and again it is subdivided into many layers like hypodermis, which is present inner to the epidermis, and it is collenchymatous in nature in case of dicot stem. And next to a hypodermis, general cortex is present, which is parenchymatous in nature, and they have clear intercellular spaces between them. Next, inner to general cortex, endodermal layer is present, which is unilayered, that means single layered, and barrel shaped and compactly arranged parenchymatous tissues. They store starch, so called as starch sheath. Casparian stripes are clearly visible in the endodermis region. It is the deposition of lignin and suberin. Several dicot stem lack those deposition, and several dicot stem also lack distinct endodermis. We can't differentiate which layer is the endodermis, which layer is the general cortex in some dicots, but generally dicot stem have endodermal layer, endodermis. Next, inner to endodermis, pericycle is present. It is the outer layer of the vascular strand or stele. It is made up of both parenchyma and sclerenchyma. Sclerenchyma lies on the outer side of the vascular bundle in the form of semicircular to semilunar patches called as bundle caps. So in case of dicot, we have vascular bundles which are arranged in a definite ring. Okay, and these bundles consist of cambium, Inner to cambium xylem is present, and outside of the cambium is consist of phloem. And these bundle caps which are associated with this phloem is called as hard bast, and it is sclerenchymatous in nature. And the region of pericycle which are present here, that means outer to the medullary rays, are called as parenchymatous pericycle. It consists of parenchyma cells. These are medullary rays. That means generally the pericycle is heterogeneous in nature. It consists of both sclerenchyma and parenchyma. Near this phloem we have sclerenchymatous pericycle called as bundle caps or hard bast. But outer to the medullary ray, because medullary rays are present in between the vascular bundles. We have vascular bundles, and in between two vascular bundles we have vascular rays, and outer to the vascular rays we have parenchymatous pericycle. So generally we have heterogeneous pericycle, but in case of cucurbita, pericycle is homogeneous. It is completely sclerenchymatous wavy layer. They do not have any parenchymatous pericycle. And sclerenchymatous pericycle provides mechanical strength to the young stem, and parenchymatous pericycle helps to store food. Okay. Next, inner to the pericycle layer, as I have already told you, vascular bundles are present which are collateral because phloem and xylem are jointly present, and phloems are present outside to the xylem, and these are open vascular bundles because they have cambium layer. Cambium layer, when cambium is present between xylem and phloem, that is called as open type of vascular bundle, open to the secondary growth, because cambium helps in the secondary growth. And these vascular bundles are arranged in a ring. In case of monocots, they are scattered throughout the stem. Each vascular bundle is formed of phloem or bast, then cambium, then xylem or wood. Phloem is external and have seed tubes, companion cells, phloem parenchyma, and it is meant to conduct the prepared food material from leaf to the storage organ. Phloem is meant for transporting food materials. Next, cambium. It is a thin walled tissue lying between phloem and xylem, and the cells are arranged in a radial row which are rectangular in shape, and cambium is responsible for secondary growth. The secondary growth starts from those cambium ring. Okay. Next, inner to cambium, xylems are present, and xylem consists of vessels, tracheids, wood parenchyma, and those are meant for conducting the water. Xylem is meant for conducting water. So tracheids, wood vessels which are large, thick walled and lie in a few radial rows. Smaller vessels towards the center from protoxylem, and bigger one lying away from the center form metaxylem. So some xylem which are newly formed called as protoxylem, which are present towards the center, and metaxylem present away from the center. Tracheid and wood fibers or xylem fibers are smaller, thick-walled and lignified cells lying around the metaxylem vessels. What is the meaning of lignified cells? Those cells which have lignin on their walls, lignin deposition on their wall. Wood parenchyma is the patch of thin-walled parenchyma lying on the inner side of the bundle around the protoxylem, and its cells are living. The cells of wood parenchyma are living, and these are patch of thin-walled parenchyma and present inner side of the bundle around protoxylem. And tracheid and wood fiber are thick-walled and lignified cell present around the metaxylem vessels. So It is all about the vascular bundles, and vascular bundles consist of xylem, phloem and cambium. Okay, so these are about different layers which are present in a typical dicot stem. So after this you can also add one example, the layers present in a sunflower stem or in a cucurbita stem. So after the general discussion or general description of a typical dicot stem, you can also add one example. Okay, so here I conclude my lecture video. I hope you really enjoyed this, and if you liked it, please hit the like button, please share with your other friends, and please subscribe to my channel to get more videos like this. Okay, thank you.